Okay. Hi everyone. I hope I am audible. If someone can respond once. Hello. Can anybody respond if I am audible and my screen is visible? Okay. Then we'll start now. So, hello everyone. Today is the 7th problem solving session for the course Biochemistry. The course instructor is Professor Swagata Dasgupta of IIT Kharagpur and I am Shrija Ghosh, PMRF from IIT Kanpur. I'll be helping you solving the assignment questions of week 7. So, week 7 is all about lipids and membrane structures, and then introduction to membrane transport, how molecules are transported across the membrane using different membrane proteins. So, we'll study about all that. We'll solve as many questions as possible on the topic, and feel free to ask any questions whenever you have any doubt. Okay, you can either unmute your mic or you can write in the chat box, whatever is suitable for you. So, we'll start with the class now. Just a second. Okay. So, starting with the first question. Okay. Cholesterol is a 27 carbon compound that has a hydrocarbon tail, a central sterol nucleus made of 4 hydrocarbon rings forming a hydrophobic ring system and a hydroxyl group. Cholesterol is expected to insert into the bilayer membrane with its W or blank oriented towards the dash, that is X, and the dash adjacent to the dash of phospholipids. Okay, so you have to fill in the blanks for 4 things. Which group is oriented towards which phase or which which location, and then with another part is oriented towards some other location in a membrane. So, we are talking about a membrane. Now, cholesterol, if you remember the structure of cholesterol, we have a hydrocarbon tail, a central sterol nucleus made of 4 hydrocarbon rings, and a hydroxyl group. So, now we have to say whether the hydroxyl group is there or the hydrophobic ring system is there and towards which part. Okay. So, we will go through the options once. Option A, hydroxyl group is oriented towards the aqueous phase and the hydrophobic ring system is oriented adjacent to the fatty acid chains. Option B is hydroxyl group is oriented towards the fatty acid chains and hydrophobic ring system is adjacent to the glycerol group. Option C is hydrophobic ring system is oriented towards aqueous phase and hydroxyl group is adjacent to the fatty acid chains, and option D is hydroxyl group oriented towards aqueous phase and glycerol groups are adjacent to phospholipids. So, which would be the correct answer? Anybody wants to answer this question? What would be the correct answer for this? Whether the hydroxyl group will be towards aqueous phase or fatty acid chains, or it would be hydrophobic systems. towards the aqueous phase or towards the fatty acid chains? What will it be? So, if you remember about polar and non-polar environments, we talked about it. So, polar compounds like to be in polar environments and non-polar compounds like to be in non-polar environment, right? So, if you remember the basic structure of a membrane, like we discussed in brief in the previous sessions also, membrane has a polar head group and a non-polar tail, right? So, if cholesterol is present inside the membrane, so it cholesterol will also face its polar groups towards the polar or the aqueous part of the membrane, and the hydrophobic tail of the membrane will be adjacent to the hydrophobic part of the cholesterol, right? So, if I just draw a diagrammatic representation of membrane, this would be the so this would be the polar head group, and these are the fatty acid chains that are there, okay, or the non-polar part. So, this is the polar part and this is the non-polar part, right? So, non-polar and this is polar. Now, in cholesterol, they have said that it has the hydrocarbon ring system and hydrocarbon ring system and then a hydrocarbon tail. So, all these hydrocarbon tail, hydrophobic ring system, these will be hydrophobic, or we can say they are non-polar, okay. So, where will this part will be? This will be towards the non-polar or the fatty acid chains. So, hydrophobic ring system will be towards the fatty acid chains. Now, hydroxyl group. What is hydroxyl group? Hydroxyl group is OH, right? So, hydroxyl group will be towards the aqueous phase or the polar part. So, this is the polar part. Hydroxyl group is polar. This is polar, right? So, hydroxyl group will be towards aqueous phase. So, the correct answer is option A, that is cholesterol is expected to insert into the bilayer membrane with its hydroxyl group oriented towards the aqueous phase and the hydrophobic ring system adjacent to the fatty acid chains of the phospholipids. Okay, this is clear. So, this is the cholesterol structure. So, if you see in the question, it is said it is a 27 carbon compound. So, do we have 27 carbons here? The carbons are numbered. So, we have 27 carbons here, or you can see the molecular formula also. So, it's a 27 carbon compound. The molecular formula is also very important. In a lot of exams, they directly ask which of the following is the molecular formula of cholesterol. So, you should remember this: C27H46 and a single O. There is only one hydroxyl group OH, so that one O is there. Then the whole hydrocarbon chain, this is the hydrocarbon chain and this is the ring system. This is whole part is hydrophobic in nature, okay, only this part is hydrophilic in nature. Now, since this is hydrophilic in nature, this part is called the polar head group, okay, polar head. So, this will interact with the aqueous medium, okay, this part can interact with aqueous medium, and this whole rest of the part will interact with fatty acid chains in the membrane, okay. So, what is this? The ring system, the 4 rings fused together. If you see here, you have A, B, C and D, 1, 2, 3, 4. So, 4 rings are fused together. D is the 5 carbon ring, rest all are 6 carbon rings. So, these 4 rings are fused together. This is the sterol nucleus. This is called sterol nucleus. Basically, this is the basic structure of sterol, okay. Then from this fourth ring we have the chain attached to it, that and the difference in these chains makes different kinds of sterols. So cholesterol, ergosterol, we'll see in upcoming slides that there are different types of sterols, and the difference is mainly in this hydrocarbon chain or the alkyl chain. So this is called alkyl chain. Okay. Okay, so we have the sterol nucleus, we have the hydroxyl group, and we have the alkyl chain. So you have cholesterol has 27 carbons. We have here cholesterol has 27 carbons. We saw 27 carbons. We saw hydrocarbon tail. We saw the sterol nucleus and sterol nucleus that are made of the ring 4 fused rings and the hydroxyl group. Okay, so this is the structure of cholesterol, and the structure and the molecular mass of cholesterol is important. You should remember, you should try to remember it. Okay, this question is clear. Okay, screen is not visible. Is it still not visible? Hello? Hello, is my screen still not visible? Okay, just give me a second. Is it visible now? Okay, I hope it is visible. So just go through the question again. So the question was, cholesterol is a 27 carbon compound that has the hydrophobic hydrocarbon tail, a central sterol nucleus made of 4 hydrocarbon rings forming a hydrophobic ring system and the hydroxyl group. Cholesterol is expected to insert into the bilayer membrane with its dash oriented towards dash, and the dash is adjacent to the dash of phospholipids. Now if you remember about polar and non-polar, I told you that the membrane has a polar head group and the non-polar fatty acid chains. So in cholesterol also, whatever will be the polar group, so out of the hydrocarbon tail and the hydrophobic ring system and hydroxyl group, only hydroxyl group is the polar. What is hydroxyl group? It is the OH. Okay, so it only this part is the polar part. So this will interact with the polar or the aqueous medium. Rest everything, the hydrophobic ring system, they will interact with the fatty acid chains that are basically non-polar. Okay, so the correct answer is option A. So I was showing just a second. Yeah, is the structure visible? Is the structure of cholesterol visible to everyone? Please respond. Okay, okay, now the screen is visible, that means. Okay, so so I was showing you the structure of cholesterol. So in the question it is written, it is 27 carbon compound. So this is the molecular formula of cholesterol. This is actually important. In a lot of exams they directly ask what is the molecular formula of cholesterol, so you should remember this. And if you see in the structure also, you can see 27 carbons. Okay, 27 carbons. The numbering of carbons are little different than the usual nomenclature, so you should see how the numbering has been done. And this is the polar head, that is the hydroxyl group that it has. Only this part is polar, rest the whole part of the cholesterol is non-polar and it interacts with the non-polar side of the membrane, that is the fatty acid chains. Okay, it interacts with the fatty acid chains. This 4-ring system, the A, B, C and D, there are 4 rings that are fused together. They are called the sterol nucleus. Okay, they are called the sterol nucleus, and the hydrophobic, so this part, this is also hydrophobic, this is a hydrocarbon chain. So single bonded carbons attached together, so this is the alkyl chain. Okay, so this is the sterol nucleus, the basic structure of sterol. This is there in all types of sterol. The changes in the alkyl chain makes different types of sterol in present. Okay, so so you have the polar head group that interacts with the aqueous phase and the rest of the ring system that interacts with the fatty acid chain. Okay, this question is clear. We will move to the next question now. Question number 2. Given is the structure of a hypothetical fatty acid, the correct nomenclature of the given structure would be. So what would be the nomenclature of the given structure? So I would like everyone to count the number of carbons themselves and tell me the correct answer. It is not very difficult. You just have to count the number of carbons and number of double bonds present. Based on that, try to find the correct answer. Even if you are not able to answer it correctly, I want everyone to guess once, because these type of questions are actually calculative. So if you don't try it, you won't learn how to write the names. So everyone try to answer. What would be the correct answer to this? Okay, we have one answer, option B. Okay, let's see now. I also don't know the answer. We will calculate the answer. Okay, so we have option B. Let's see if that's the correct answer or not. So now how to name any fatty acid? So we have to start counting the number of carbons. So we will start from here. So this will be the first carbon, then second, third, fourth, fifth, sixth, seventh, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 and 20. Okay, so we have 20 carbons. So the first number that is shown in the nomenclature, that is the number of carbons. So number of carbons is 20, that is correct. Now comes the number of double bonds. Okay, so number of double bonds, how many double bonds do we have? This is first, second, third, fourth and fifth. We have 5 double bonds. Okay, so we have 20 carbon, 5 double bonds. Now the number after delta that is shown here, it is basically from which carbon the double bond is starting. Okay, so in this case the double bond is starting from the 5th carbon. Okay, so 5 double bonds are there and the double bond is starting from 5th carbon. So that means the correct answer is option D. So do you understand? So if the double bond was starting from this place, the answer would have been option B. But right now the double bond is starting from the 5th carbon, right? So 1, 2, 3, 4, 5. After fifth carbon we have the double bond between 5 and 6. Then it will go after 3 carbons. Okay, so after fifth it will be always 5 plus 3, 8, then 11 and so on. Okay, so 5 and 6, then 8 and 9, then 11 and so this is a hypothetical structure. So generally if you make a actual, like if you see an fatty acid which has double bonds, they are generally separated by 3 carbons. Okay, so basically the starting number from which carbon the double bond is starting, that is mentioned after the delta. Okay, so if I am giving you a fatty acid written like this, that means it has 20 carbons, number of carbons. This is the number of double bonds. And this shows the nth carbon from which double bond starts. Okay, okay, so do you understand? If you have a nomenclature like that, number of carbons is the first thing. So you were right about option B, 20 was correct. Number of double bonds is the second thing. So 1, 2, 3, 4, 5. So even 5 was correct. The only thing you were wrong about was from which carbon the double bond is starting. So if you start numbering from here, I think you might have started numbering from here, and that's how you have gotten 3. But you always, in this delta format, when you are writing, you always have to start counting from the carboxyl group, carboxyl carbon. Okay, so since it's a fatty acid, acids have this COH group, you always have to start counting. The first carbon will be the carboxyl carbon. Okay, so you have to do the counting correctly, only then you will get this number correct. Okay, this is fine. So I hope everybody understood how to give names to fatty acid. Like if I draw a random fatty acid, you can give the name of that fatty acid. Okay, you just have to count the number of carbons, the number of double bonds, and then from which carbon the double bond is starting. Okay, chalo. So we will move to the next question now. Question number 3. So steroids are oxidized forms of sterols. Steroids have a dash. Option A, sterol nucleus with 2 alkyl chains attached to the ring D of cholesterol. Sterol nucleus with 2 CH3 groups or methyl groups between the C and D rings and A and B rings of the cholesterol. Sterol nucleus with no methyl group between C ring and D ring of the cholesterol, and sterol nucleus but lack the alkyl chain attached to the D ring of cholesterol. Okay, so what would be the answer for this? So what would be the structure of steroid? I have shown you the structure of cholesterol. So you had the sterol, you know what a sterol nucleus now. So anybody can answer this? The correct answer would be, the correct answer is option D. Okay, sterol nucleus but lack the alkyl chain attached to the cholesterol. So you see, this was the cholesterol structure, right? So I told you this is the steroid nucleus or sterol nucleus. Okay, so this part is the sterol nucleus. Okay, now from this, if I remove this hydrocarbon side chain, if I end the structure here at the pink part, if I end the structure, what will you have? You will have the structure of steroid. Okay. So difference between steroid and sterol is basically the presence of, so this whole thing, the whole thing that I have boxed it out, this will be steroid. Okay. And if I attach this hydrocarbon chain, it will become a sterol. So sterols are basically oxidized forms of steroid. Okay, steroids are oxidized forms of sterols. Okay, this is clear. So steroids have a sterol nucleus, but they lack the alkyl chain that is attached to the D ring of the cholesterol. So you remember, this is the fourth ring, the last ring, that is a 5 carbon ring, that is the D ring. Okay, this is 1, 2, 3, 4, 5 carbon ring. This is the D ring. From this the alkyl chain is attached. If I remove this alkyl chain, it will be the steroid. Okay, I hope this is clear to everyone. Okay, moving to the next question. Given figure shows a schematic representation of the various types of membrane proteins. Identify the types of proteins labeled as X, Y and Z. Okay. So we have a schematic here. Now you have to tell what is X, what is Y and what is Z. This is very simple. So option A is X is peripheral, Y is integral and Z is lipid anchored. Okay. B is X is integral, Y is peripheral and Z is lipid anchored. C is X is peripheral, Y is lipid anchored and Z is integral. And fourth is X is lipid anchored, Y is integral and Z is peripheral. So what would be the correct answer here? This is very simple. So anybody can answer this. You see where the protein is, whether it is integrated in the membrane or it is on the periphery of membrane. So you understand the terms periphery and integral, right? So based on that you can easily see which protein, which structure represents what. Anybody? What would be the answer for this? This is very simple. Where is the peripheral protein? What do you mean by periphery? Periphery means on the surface, right? Periphery means on the surface. And integral means it is embedded inside. Okay. So periphery means on the surface and integral means it is embedded inside. So now if you see what is on the surface, X is on the surface, right? So X will be what? X will be peripheral. So option A and option C both has X as peripheral. Now what is Y? Y is embedded in the membrane, right? So integral will be into the membrane or embedded in the membrane. Okay. So this blue thing that you see here, this is the membrane, basically membrane bilayer. Okay. So X is peripheral protein, Y is integral and Z is something called lipid anchored. So it is not a integral protein, but it is anchored by the lipid. So this is the membrane, right? So membrane has the polar head group and the non-polar chain. So this part that you see here, this part is anchored by lipids. So this is the non-polar part of the membrane, the inner part, the light blue part is the non-polar part. So lipid is anchoring that part and the rest of the protein is outside on the periphery. Okay, so Z is the lipid anchored protein. So we have peripheral proteins, integral proteins and lipid anchored proteins. So these are three types of membrane proteins. Okay, so we will see where these membrane proteins are important. They are important in membrane transport, different types of membrane transports. So these membrane proteins, there are three types. They can either be on the surface of membrane, they can be embedded inside the membrane, or they can be just anchored to the membrane with a lipid, by a lipid chain. Okay, so lipid anchored. This is clear? So this was question number 4th. Okay, the correct answer is option A. Now we move to next question, question number 5. Which property can be shared by the given lipid and a terpene? So which property can be shared by a given lipid? This is the lipid that is given here, and a terpene. Both can contain isoprene, both can form micelles, both can contain a saturated fatty acid, and both can be very hydrophobic molecules. So what would be similar between a lipid and a terpene structure? So terpenes are actually class of isoprenes. So terpenes have isoprene, but there is no isoprene structure in this whole lipid, right? So there is no isoprene structure in this whole lipid, so this will not be the property that both are sharing. Okay, both can form micelles. Now you know lipids can form micelles. What are micelles? They are basically amphipathic molecules. So we have lipids that has a polar head group and a non-polar chain, right? So a polar and a non-polar part. When these lipids are in a solution that is aqueous, so what will happen? All the polar parts, they will be on the surface and all the non-polar chains will face inwards and form a circular or spherical structure called micelles. Okay, sorry. So polar part, so this is the outer environment is aqueous. Okay, the outer environment is aqueous here. So if the outer environment is aqueous, if the outer environment is aqueous, so polar head groups will face towards the aqueous part and the non-polar chains will face towards each other. So non-polar can face non-polar, so it will make a circle basically. Okay, a sphere. Imagine a ball where the surface of the ball is polar and the inner core is non-polar. Okay, so that would be a micelle. So any lipid, if you see, these are the long chains that you see, they are the hydrophobic part of the lipid. Okay, so these are long hydrocarbon chains, so they are very hydrophobic. Okay, so they will be inside of the micelles and the outer part will be polar. Okay, so they can form micelles. Same with terpenes, since terpenes are also amphipathic molecules, they also have polar and non-polar parts, so they will also form micelles. So both can form micelles. This is a correct statement. So both can form micelles. Now both can contain a saturated fatty acid. So we have saturated fatty acids. These are the fatty acid chains. These are the fatty acid chains. Okay, so both of them has fatty acid chains. This lipid has a fatty acid chain, but terpene does not have a fatty acid chain. So both do not contain, only lipids contain saturated fatty acid chains. Now both can be very hydrophobic molecules. That is true because I told you they are forming micelles. That means they have the hydrophobic properties. That's why only they are forming micelles. So second and fourth are correct. So correct option is C. Second and fourth statements are the properties that are shared by both a lipid and a terpene. Okay. So you have to understand the concept of micelle formation, what is hydrophobic, what is hydrophilic, how the moieties are being how the moieties are interacting. The hydrophobic is interacting with hydrophobic and hydrophilic is interacting with hydrophilic, basically like so polar will like the polar environment, non-polar will like the non-polar environment. Okay. So I hope this is clear. Okay. Is there any doubt till this part? If you have any kind of doubt, ask me now so that we can move on to the next question. Okay, so question number 6. Palmitic acid and palmitoleic acid are two fatty acids whose structures are given, provided are the melting curves for the two given molecules. So we have palmitic acid structure here and palmitoleic acid structure here. Okay. And the melting curves are provided. This is one melting curve, this is the second melting curve. Assign the curves for as that for palmitic and palmitoleic acid. So you have to, based on the structure, you have to see whether this curve will be assigned to palmitic acid or this. Okay, so X and Y, two curves are labeled. The blue curve is X, the red curve is Y. So whether X curve is for palmitic acid and Y is for palmitoleic acid or vice versa, you have to answer this. So how will we see? So the melting curve is dependent on what? It is dependent on the structure, right? How it is dependent on the structure, we will see. So if we have a straight chain fatty acid, what do you mean by straight chain? There are no bends in this. All the bonds are single bonds. So it's a straight chain, right? So the molecule is free to rotate on its axis over like it can rotate about any of these bonds, right? So if I have a single bond and two carbons attached from a single bond, the molecule can rotate any way it bonds to. So if you remember the structure of peptide, like a peptide bond, it was a planar bond, right? So there was a rotation about that planar bond. Okay, when that rotation was restricted, how was in which amino acids we saw the restriction in? We saw the restriction in the rotation. The rotation was restricted when there was a big group that was hindering the motion. So if I am saying that there are two objects that can freely rotate, now I put obstruction in front of one object. Now this cannot rotate as freely as it was rotating, right? So similarly, if you see the structure here for palmitic acid, the structure is very straightforward. It's a straight single bonded carbons attached together. So it can have a free rotation. So it would be a very sturdy structure, right? Because to break down this structure, there is no obstruction. So you will have to break each and every bond. So you will need more energy. Now in palmitoic acid, since there is one double bond, because of this double bond there is a kink in the structure. The chain is not straight now. So if you have, let's say these fatty acids are in a membrane. Okay, so with a polar head group, there is a palmitic acid and a palmitoleic acid. Okay, So palmitoleic acid has a kink here, right? And palmitic acid is straight. So if you see, if I have to break this, it already has a bend. So it would be easier for me, less energy would be required for me to break this off. So if I give you a pencil which is half broken, and if I give you a straight whole pencil, what would be easy to break? A pencil that is half broken, you just have to bend it further to break it, right? If you have a sturdy pencil without any cracks, it would be very difficult for you to break it, right? So that means more and more energy is required to break palmitic acid. That means higher temperature would be required for it to melt. Melting means when it denatures. Okay, so if I am heating a compound, it will denature when it will reach a certain temperature. Okay, and that temperature is the melting temperature. Okay, so for palmitic acid to break or to denature, it will require a higher temperature. So, and palmitoleic acid, since it already has a kink or a crack or a break, so it would require a very less temperature to, a very low temperature to break it off. So that means curve X is denoting palmitoleic acid and curve Y is denoting palmitic acid. So now the correct answer is option C. So do you understand how one kink is introducing such a huge difference in the melting temperature? So now if I introduce one more double bond here, there will be a further kink. So the molecule will bend like, right now the molecule is like this, then the molecule will become like this. So it would be further easier to denature it. Okay. So more the number of double bonds, more the number of kinks will be there. Okay. So higher double bonds will lead to more kinks, and more kinks means lower melting curves or TM, lower melting temperature. Okay, so higher number of double bonds, more kinks, so lower temperature, and the opposite, lower number of double bonds, that means less kinks and higher TMs. Okay, so the correct answer is option C. I hope everyone understands. I hope this is clear to everyone, right? If this is clear, we will move to the next question. A cerebroside is a dash with a monosaccharide such as a glucose or a galactose as the polar head group, whereas ganglioside is a ceramide with a polar head group that is a dash including the acidic sugar derivative sialic acid. These cerebrosides and gangliosides are collectively known as dash. Identify P, Q and R. So let's see the options. The option A says P is sphingolipid, Q is complex oligosaccharides and R is glycosphingolipids. Option B says P is glycerophospholipid, Q is disaccharide and R is glycosphingolipids. Option C says P is sphingolipid, Q is monosaccharide and R is glycolipids. And option D says P is glycerophospholipid, Q is complex oligosaccharide and R is glycolipid. So what would be the correct answer here? So what would be the correct answer here? We have cerebroside is a dash. Cerebroside is a dash. So whether cerebrosite is a sphingolipid or a glycerophospholipid, it is a sphingolipid. Okay, cerebrosite is a sphingolipid with a monosaccharide such as glucose or galactose as the polar head group, whereas ganglioside is a ceramide with a polar head group. That is, what is the polar head group in ganglioside? In ganglioside, the polar head group is complex oligosaccharides. Okay, in cerebroside it is a monosaccharide, but in ganglioside it is complex oligosaccharides, including the acidic sugar derivative sialic acid. These cerebrosides and gangliosides, they are what? They are basically glycosphingolipid. Why the term glyco is there? Glyco means sugar. Okay, so the polar head group has sugar in it, so it is glyco, and they are sphingolipids. Okay, now we will see what the sphingolipids are. So the correct answer is option A. Okay, cerebroside is a sphingolipid with a monosaccharide such as glucose or galactose as a polar head group, whereas ganglioside is a ceramide with a polar head group that is a complex oligosaccharide, including the acidic sugar derivative sialic acid. These cerebrosides and gangliosides are collectively known as glycosphingolipids. So this is a sphingosine molecule. What's a sphingosine? Basically, this is the backbone structure of any sphingolipid. Okay, so sphingolipids, the term sphingo is coming from sphingosine. So sphingolipids have this backbone structure. So if you see this backbone structure, there is a long hydrocarbon chain present, there is an amino group present. This amino group will be esterified into it will be esterified with a fatty acid chain. So this part will again have a long hydrocarbon chain of the fatty acid, and then this OH, this will basically become the polar head group. From this, any polar compound can attach and become a polar head group. So we will see the structure of cerebroside first. So cerebroside, it is a sphingolipid which has, so this, if you see, when a fatty acid is attached to this amino group, this part is called ceramide. Okay, so if I, instead of this amino group, if I attach a fatty acid here, okay, this will become what? This will become ceramide. Okay, so cerebroside is a sphingolipid that has this ceramide backbone structure and the monosaccharide. So here beta galactose is shown. So glucose or galactose can be present as the polar head group. So this will be the polar part, the one I am circling out. This is the polar part and this is the non-polar. So these are the non-polar fatty acid chains and the non-polar hydrocarbon tail. So polar and then non-polar chains, two chains are there. Okay, so this is the basic structure of sphingolipids. So cerebroside is one in which this polar head group is a sugar, that is a monosaccharide. Okay, monosaccharide. So glucose or galactose, these are monosaccharides. Now comes another sphingolipid, ganglioside. So ganglioside also has this ceramide structure. So if you see here, so this carbon from which the hydrocarbon chain, this is this hydrocarbon chain that you see here, this hydrocarbon chain that you see here. Okay, this hydrocarbon chain is shown here with the black part. Okay, okay, this is the hydrocarbon chain. Now if you see from this hydrocarbon chain, this carbon is there from which the amino group is there. So this is the second carbon from which the amino group is there. From this amino group there is a fatty acid attached, a stearic acid. It's a fatty acid that is attached. Then comes the third carbon from which the polar head group. So this is the polar head group, and polar head group is a complex oligosaccharide. So complex oligosaccharide means multiple sugar molecules joined together to form the polar head group. So, it has a bigger polar head group. Okay. For cerebroside, it has a small head, but for ganglioside, it has a bigger polar head. Okay. So, ganglioside and cerebroside both are called glycosphingolipids. The term glyco means sugar. So, since both of them has sugar as the polar head group, it is called glycosphingolipids. Both of them are sphingolipids. Okay. The backbone structure is ceramide. Ceramide has a fatty acid attached to the amino group of the sphingosine. So, you have sphingosine. See here, this is sphingosine. When I attach a fatty acid to this amino group, it will become a ceramide, and if I attach the sugar to this OH, the third carbon, the OH attached to the third carbon, so that ceramide will change into a glycosphingolipid. Okay. So, if I attach a sugar to that OH, it will become a glycosphingolipid. If I attach any other molecule, it will become sphingomyelin or some different membrane structures. Okay. So, these all are basically parts of membranes. Okay. Phospholipids, sphingolipids, these are glycerophospholipids. These are basically different types of membrane components. So, membrane is composed of these different types of lipids that has a polar head group and a non-polar chain. This is clear? Okay. If this is clear, we will move ahead to the next question, question number 8. The detailed representation of the fluid mosaic model is shown here. Identify the unknown element P, Q and R from the above image. So, you can see the image. This is the fluid mosaic structure of the protein. So, we have to name what is P, what is Q and what is R. So, we already did a similar question in the previous slides. So, tell me what is P, what is Q and what is R. Option A is P is integral protein, Q is cholesterol and R is protein channel. Option B is P is surface protein, Q is cholesterol, R is protein channel. Option C is P is integral protein, Q is glycolipid and R is glycoprotein. Option D is P is surface protein, Q is glycoprotein and R is glycolipid. So, what would be the correct answer with what is P? P here is, you can see it is embedded in the membrane. So, what did I tell you? What would be the correct answer? Come on. Come on, what would be the correct answer here? I told you already, if it is embedded in the membrane, it is called integral protein, right? So, P is integral protein. If it is on surface, if it is on the surface, it is called peripheral proteins. If it is embedded into the membrane, it is called integral protein, right? So, P is integral protein. Now, what is Q and what is R? So, Q is basically the cholesterol. So, we have been studying the structure of cholesterol and sterols. So, Q is here, you can see the cholesterol molecule. So, the head group, the polar OH part is outside and the hydrophobic chain will be towards the inner side. So, cholesterol, and why is cholesterol embedded in the membrane like that? Cholesterol basically regulates the membrane fluidity. Okay, cholesterol is responsible for regulating membrane fluidity. Okay. So, if we have a very low temperature or a very high temperature, so your membrane structure should not disrupt, right? So, the membrane should be fluid enough so that even if it is very low temperature, even if it is at a very high temperature, it functions properly. So, it should not happen that if our cell or any membrane is at a very high temperature, it disintegrates. It should not disintegrate, right? So, for that purpose, cholesterols are there into the membrane structure, so that it provides a certain rigidity to the membrane and it helps regulating that fluidity. So, it makes the membrane fluid at low temperature and it controls its fluidity at a higher temperature. Okay. Then R is protein channel. So, you see there is a channel formed between this structure. So, these are basically you have the polar amino acids lined and then the non-polar amino acids are towards the outer part. So, you have bilayer kind of structure. So, imagine a thick pipe or a straw. Okay. So, this outer surface of the straw would be non-polar, sorry, would be yeah, the outer surface of the pipe would be non-polar and the inner surface, the hollow part, that would be lined with polar molecules so that it can facilitate the movement of polar molecules. Okay. So, they form channels that has, so this part would be, the inner part would be polar and it can help passing or transporting polar molecules across the membrane, help transporting polar molecules. Okay. So, these are channel proteins. Okay, protein channels. Fine, they are making a channel. Glycolipid, I already gave you an example of glycosphingolipid. So, any lipid from which a sugar molecule is attached, it is glycolipid. Any protein in which a sugar molecule is attached, it is called glycoprotein. Okay. So, concept of peripheral protein, integral protein, surface protein, cholesterol, channel proteins, this should be clear. Okay. Fine. So, I will move ahead. The correct answer here is A, option A is the correct answer. P is integral, Q is cholesterol and R is the protein channel. Okay. So, moving to the next question, question number 9. Which of the following acts as the barrier to most water soluble substances? Okay, which of the following acts as barrier to most water soluble substances? Phospholipids, hydrophilic heads, nucleus and DNA. Deoxyribonucleic acid, this is DNA. So, which of the following acts as a barrier to most water soluble substances? So, what would prevent water soluble substances from moving? What would be the answer? Come on, this is a very easy question. I want the answer from you guys. You all have to respond, otherwise there is no point of having these problem solving sessions. These sessions are for you all. You have to ask questions, you have to respond. What would be the answer to this? I have told you polar molecules like the polar environment and non-polar molecules like the non-polar environment. So, water soluble substances are polar, right? Water soluble substances means they are polar. So, if they are polar, what would they like? They will like polar. So, what would be a barrier to them? The barrier would be non-polar. So, which of the following is a non-polar barrier? a hydrophilic head, whether it would be a non-polar barrier? No, it is hydrophilic, so this is polar, it cannot hinder. Nucleus and DNA, they won't act as a barrier to water soluble substances because DNA itself is water soluble. Nucleus has a water soluble environment. So what would be the barrier to water soluble substances? Phospholipids. Okay, phospholipids. What are phospholipids? So I told you phospholipids are basically again parts of membranes only. So you have the lipid structure, the fatty acid chains attached. So if it is a glycerophospholipid, so it will have the glycerol, the basic framework of glycerol, and from two of the carbons there will be fatty acid chains attached, and from the third carbon there will be a polar head group, and this carbon will have a phosphate also attached. Okay, so there will be a phosphate group PO4, and there will be a fatty acid and a polar head group. So the polar head group will interact with the aqueous phase and the non-polar fatty acid will interact with the non-polar environment. So if these phospholipids make the membrane structure, it will form a barrier because water can interact with this part, but water cannot interact with this part of the membrane. So water soluble substances, they cannot pass through that. They need certain helper proteins, the membrane proteins that we were seeing. Basically those membrane proteins are helping these substances to pass through the phospholipid membrane. They cannot cross the membrane on their own. This is clear? So the correct answer is option A. Phospholipids acts as barrier to most water soluble substances. Okay. Moving to the next question, question number 10. Identify the states A and B given that the diagram represents the membrane fluidity and how it varies with temperature. So this represents the membrane fluidity. We have a state A. When we heat this, we get the state B. When this B is again cooled down, we get back the state A. So these are basically membranes. So these are the polar head groups. This is the polar head group. This is the polar head group. And this is the fatty acid chains or the hydrophobic chains. Okay. So now when heating it, they are getting disrupted, and while cooling it, they are in the perfectly ordered structure. So what is the state A and what is state B? What would be the answer for this? So the correct answer is option D. So state A is gel and state B is liquid crystal. Okay, liquid crystal. So what is happening? This is gel, or this is the solid crystalline form. Okay, so everything is perfectly ordered. Okay, when we are heating it, these fatty acid chains, they will get disrupted because they will start denaturing. You saw the melting curve of the fatty acid, right? So at certain temperature, different fatty acids will start melting. Okay, so they will start disrupting and they will make the whole membrane structure, the ordered structure, little fluid. Okay, so this will be fluid, whereas this will have no fluidity or very low fluidity because this is very rigid. This will be a rigid structure. Okay, so from rigid structure, when we are heating it, it becomes fluid, and when we cool down, it again becomes rigid. So remember, I told you about cholesterols being part of these lipid bilayers. So what happens cholesterol molecules are present between few lipid molecules to maintain this rigid to fluid dynamics. Okay, so at a lower temperature the structure becomes rigid. It would be very difficult for membranes or cells to function with a rigid membrane, right? It needs a certain amount of fluidity for the things to pass through and the membrane proteins to fit properly and move, change its conformation and so on. So the cholesterol molecules basically help the membrane to stay in this liquid crystal form even at low temperature. Okay, so if cholesterol is present, the membranes will be in this liquid crystal form even at low temperature, even when you cool it down. Okay, so from this highly ordered structure, from this crystalline or gel structure, we get a liquid crystal or a fluid structure. Okay, so correct answer is option D. This is clear? Next question, question number 11. What do B, C and D represents in the figure? So here we have three types of transporters shown. What are these? So in first case we have only single arrow going from left to right. In the second case we have one arrow going from left to right and second arrow going from right to left. In the third case we have both the arrows going from left to right. So what would it be? Option A is B is symport, C is uniport, D is antiport. Option B is uniport, antiport and symport. Option C is B antiport, C symport, D uniport, and option D is B uniport, C symport and D antiport. So what would be the correct answer here? The correct answer would be, what is B? B is uniport. Uni means one, right? So uni means one. So here we have only single molecule passing from left to right. So let's say we have a molecule X. X is passing from left side to the left side of the membrane to the right side of the membrane. These are the carrier proteins, different types of carrier proteins. So this is a uniport type of carrier protein. Now comes C. C is antiport. By anti means opposite. So you remember parallel and anti-parallel beta sheets, right? So anti means opposite. In that you had opposite strands of beta sheets, so it was anti-parallel. So similarly antiport. So antiport means opposite direction, transport in opposite direction. So one molecule will pass from left to right, at the same time another molecule will pass from right to left. So let's say H plus is passing from left to right. So at the same time a molecule X will be passed from right to left. So opposite transport. Okay. So antiport means opposite transport, uniport means single transport, uni means one, so single transport, and the last is symport. So sim means it's opposite of anti basically. So anti means opposite, sim means in the same direction. Okay. So in the same direction. So two molecules, if H plus and let's say Na plus, they are moving together. So whenever H plus will move from left to right, at the same time Na plus will also move from left to right using this symport carrier protein D. Okay. So the correct answer would be option B, that is first is uniport, second is antiport and third is symport. Okay, uniport, antiport and symport. This is clear? This is clear, I hope. Okay. Okay. So moving to the next question, sphingolipids are derivatives of the lipid sphingosine, which has a long hydrocarbon tail, a polar domain that includes an amino group. Glycosphingolipids, a derivative of sphingolipids, is a combination of option A, glycerol with 2 galactose residues, option B, ceramides with 1 or more sugar residues, option C, sphingosine with galactose and ceramide, and option D, sphingosine with glucose. So, sphingolipids, I showed you the structure of sphingosine, right? So, sphingosines, it is a derivative of sphingosine. In the sphingosine, we have an amino group which attaches a fatty acid chain and becomes ceramide. Then, it already has a hydrocarbon tail, the sphingosine. Then, the polar domain, it will include, so one polar domain has the amino group, one polar domain has the OH group. So, OH group will become the polar head and the amino group will be connected to a fatty acid to become a ceramide, okay, and then that ceramide can be further modified into different types of sphingolipids. So, what is glycosphingolipid? So, I told you what does glyco means. Glyco means sugar. Okay, so any type of sugar, not just galactose or glucose. Okay, any type of sugar. So, ceramides with 1 or more sugar residues. This would be the correct answer. Ceramides with 1 or more sugar residues are basically glycosphingolipids. Okay, so option B is correct. So, this was the sphingosine structure I showed you. So, this is sphingosine. So, you have a carbon with the, so this will make the polar head group. This has the amino group. So, this amino group can further get connected to a fatty acid chain. So, this will be a fatty acid chain. So, now the sphingosine already had a hydrocarbon chain. Now, it attaches to a fatty acid chain. So, there are 2 hydrophobic chains. So, this becomes a ceramide structure. Now, in the ceramide structure, if I attach another polar compound to this OH, so let us say I attach a sugar, it will become a glycosphingolipid. Okay. If I attach a phosphate group, it will become a phosphosphingolipid. Okay. So, whatever is attached to this OH, based on that the nomenclature will go. So, glyco means sugar. Okay. So, any sugar attached to the ceramide will make it glycosphingolipid. Okay. So, any sugar molecule that is attached to the ceramide, the polar head of the ceramide, that will make it a glycosphingolipid. So, this part will be polar head group and this part will be non-polar side chains. Okay. So, cerebroside, I showed, I have told you cerebroside and ganglioside, they are 2 examples of glycosphingolipids. Okay. Gangliosides has complex sugar oligosaccharides or sugar polar head group. So, multiple sugar molecules connected together, and cerebrosides has monosaccharide like galactose or glucose. Okay. So, the next question, question number 13. Consider a cell membrane with a potential difference across it. Given that delta mu is equal to minus 9000 joules per mole, calculate the potential difference across the membrane V2 minus V1. R is given as 8.314 joule per Kelvin per mole. F is the Faraday's constant, that is 96,500 coulombs per mole, and temperature is given as 25 degrees Celsius. So, you have a cell membrane here, this is the cell membrane, this part is the cell membrane. So, one part has 1000 millimolar calcium concentration, second part has 10 millimolar calcium concentration. So, calcium ions are higher on first part V1 and lower on the second part. So, there will be a potential difference across this membrane. So, V2 minus V1. So, we have to calculate the potential at V2 minus V1. Okay. So, the potential difference across the membrane. How do we calculate that? So, to calculate that, we will use a formula, that is delta mu is equal to RT, R is the gas constant, T is the temperature, log of calcium ion concentration in the second chamber divided by the calcium ion concentration in the first chamber. So, lower divided by higher, plus Z, Z is the valency, F is the Faraday's constant, and V2 minus V1 is the potential difference across the membrane. Okay. So, this is the valency, this is the Faraday's constant, and this here is the potential difference. Okay. So, you have the delta mu given in this. So, delta mu is minus 9000 joules per mole. So, we have delta mu as minus 9000 joules per mole, R is given, F is given, T is given. T is given in degree Celsius, but if you see the unit of temperature in R, it is in Kelvin. So, you have, whenever you are solving any equation, you have to make sure all the units are in the same format. So, if this is joule, this is joule, then it is fine. If this was given in, let us say this was given as 9 kilojoule minus 9 kilojoules per mole. Okay. So, in that case I had to convert this into joules per mole, then only I can use this value of R with this value of delta mu. Okay. Everything should be in the same unit. So, this is delta mu is minus 9000 joules per mole, R is given, F is given, T I have converted. So, 25 degrees Celsius. So, you just add 273 plus whatever degree Celsius is there, this will give you the value in Kelvin. Okay. So, 273 plus 25 gives us 298 Kelvin. So, now just replace the values of all these things. So, delta mu is minus 9000, R is 8.314, T is 298, log of calcium ion concentration in the second chamber, that is given as 10 millimolar, and in first chamber it is 1000 millimolar. So, 10 divided by 1000 plus we have Z. Z is basically the valency. So, calcium has a valency of 2, Ca2 plus. So, Z will be 2, Faraday's constant we have given, and the potential difference. So, this is what we have to calculate, V2 minus V1, this is what we have to calculate. So, when you solve this whole thing, you will get V2 minus V1 equal to 0.0125 volts or 12.5 millivolts. So, you have to be very careful about the unit also, because all the answers are 12.5. Okay. So, you have to be careful about whether you are getting a positive value or a negative value. So, here when you solve this whole thing, you will get a positive value. Okay. You will get a positive value for V2 minus V1. Why did we get a positive potential difference? Because the concentration at V2 is less than the concentration of V1. So, the potential difference is in positive. If I will calculate for V1 minus V2, I will get a negative potential. Why? Because the concentration at V1 is already higher than the concentration of V2. That means the movement from V1 to V2 is easier. Okay. If something is higher on one side and lower on the other side, the movement will always from the higher to the lower. So that would be a negative potential. That would give us a negative potential there, so that the movement is facilitated, thermodynamically favorable. Okay. Now for V2 minus V1, it is a positive potential, and so you have to be careful about whether it is a positive or a negative value. And the second thing that you have to be careful about the unit, because if you just calculate the answer without thinking about the unit, you will get 12.5, but whether that 12.5 is volts or millivolts, okay, that you have to see. So the answer we got is 0.0125 volts. So that means if you multiply it by 1000, so it would become 12.5 millivolts. So correct answer is option A, plus 12.5 millivolts. Okay, rest of the answers are wrong. This is clear. So these type of numericals are also important in this chapter. So be careful about how to solve that, and when you are solving, be careful about the units that are given in the question. So any doubt till this question? In any of the question, if you have any doubt, please ask. If you are not asking any doubt, I will just assume that you don't have any doubt. I will just finish my session and that's it. This session is for you to ask me doubts, so it would be very helpful if you interact more. Okay. I hope everyone is understanding the questions, right? Okay. So moving on to the next question now, question number 14th. Just a second. Question number 14. Plants and fungi generally do not contain cholesterol. Instead, plants have other types of sterols such as dash, and fungi use dash as their major sterol. So identify M and N. So plants and fungus, they have different types of sterols other than cholesterol. So what is it that plants have and what is it that fungus have? So plant has a sterol called a stigmasterol, and fungus have a sterol called the ergosterol. Okay. So correct answer here is option C. So here is the structure of cholesterol, stigmasterol and ergosterol. So stigmasterol is there in plants and ergosterol is there in fungi. Okay. So I told you cholesterol is a 27 carbon compound, right? And I told you the difference in this alkyl chain. This is the alkyl chain. Difference in this alkyl chain makes different types of sterol. So you see here in the stigmasterol, the base structure, the sterol nucleus is same, the polar head group is same, but in this alkyl structure you see there is a double bond here. You see this double bond here, that is different. Then what is different? There is a ethyl group. So you see this group. So there a double bond here and a ethyl group on this 24th carbon. It makes it different from cholesterol. So from cholesterol there is a double bond between double bond between carbon 22 and 23, right, and ethyl group on carbon 24, on carbon 24, C24. Okay. So that makes it stigmasterol. What is there in ergosterol? So ergosterol, the ring structure is same. There is an additional double bond between the 7th and 8th carbon. So, double bond between 7 and 8. And what is the difference here? We have this double bond that is there in the stigmasterol between 22 and 23. So 22 and 23 also has a double bond, and instead of a ethyl group we have a methyl group here, right? We have a CH3. So instead of ethyl we have methyl group on carbon 24, right? So we have double bond between 22 and 23 both in stigmasterol and ergosterol, and a ethyl group in case of stigmasterol and a methyl group in case of ergosterol on the carbon 24. In ergosterol there is an additional double bond in the ring structure also. So this ring structure, what did I tell you, what is it called? It is called the sterol nucleus. Okay, the ring structure is the sterol nucleus where 4 rings are fused together. Okay. Okay. So this is the sterol nucleus. This part is the sterol nucleus, the polar head group, and the alkyl chain. Difference in the alkyl chain makes it different types of sterol. So this is cholesterol, this is stigmasterol, and this is ergosterol. Okay, I hope this is clear. Moving to the next question, question number 15. Which of the following best explains why the plasma membrane of the cells exhibit a negative resting potential? Okay, why the plasma membranes of the cells exhibit a negative resting potential? So option A is the membrane is most permeable to chloride ions, and the chloride gradient favors its diffusion out of the cell. Option B is membrane is most permeable to potassium ions, and the potassium gradient favors its diffusion into the cell. Membrane is most permeable to sodium ions, and the sodium gradient favors its diffusion into the cell. And the membrane is mostly permeable to potassium ions, and the potassium gradient favors its diffusion out of the cell. So what would explain the negative resting potential of a plasma membrane? The correct answer is option D. So membrane is most permeable to potassium ions, and the potassium gradient favors its diffusion out of the cell. So basically in a plasma membrane the potassium ions are more permeable than the sodium or the chloride ions. Okay, so potassium ions can leak out of the membrane via diffusion. Okay, so potassium ion will be diffused out of the cell, and the sodium ions and chloride ions will be, the diffusion of sodium and chloride ions will be slower. So there will be more positive charge going out of the cell. So that will make a negative resting potential across the plasma membrane. Okay, so is this clear? So more and more potassium are going out, but in the same proportion the chloride ions are not coming in, or the sodium ions are not going out. So there is a potential difference. There is a difference in the concentration of the potassium ions, sodium ions, and chloride ions. So there is a gradient formation that is happening, and due to that gradient formation, since the membrane is more permeable to potassium than sodium and chloride, it gives a negative resting potential to the plasma membrane. Okay, this is clear. Okay, moving to the next question, question number 16. Which of the following attributes of the membrane is most affected at the transition temperature? So which of the following attributes of the membrane is most affected at the transition temperature? Option A is size, option B is fluidity, option C is internal composition, and option D is internal environment. So what would be most affected in a membrane at a transition temperature? What do we mean by transition temperature? It is basically the melting temperature. So, from where the membrane is transitioning from one form to another. So, if you remember the ordered and the liquid crystal structure that I showed in the previous slide. So, we had so you have an ordered structure, this is the gel form and this is the liquid crystal. Okay. So, when we are heating the membrane, when we are heating the membrane, which factor or which attribute is getting affected? The fluidity is getting affected. The size of the membrane remains same, the composition and the environment remains same. The fluidity is affected. Why? Because the hydrophobic tails that the membranes have, these hydrophobic tails are basically start melting, okay, start getting disrupted or denatured. Because of the denaturing of these hydrophobic tails, what is happening? The membrane is becoming more and more fluid. So, from a ordered gel or a crystal structure, so this is a rigid structure, the membrane becomes fluid. Okay. So, the attribute that is being affected at the transition temperature. What is transition temperature? That is the temperature at which this transition is happening from one form, the transition is happening into the second form. That transition is happening at a particular temperature, that is called the transition temperature, and this transition is affecting the fluidity of the membrane. Okay, so the correct answer is option B. This is clear? The correct answer is option B. So, fluidity is affected, most affected in the membrane at a transition temperature. Now, next question, question number 17. The dash degree of unsaturation of the fatty acid of the bilayer, the dash the temperature before the bilayer gels. So, again this is about the gel to liquid crystal transition only. So, option A is greater the degree of unsaturation, lower the temperature before the bilayer gels. Option B is greater the degree of unsaturation, more the temperature before the bilayer gels. Option C is lesser the degree of unsaturation, higher will be the temperature, and then option D is less. Okay, this is wrong. So, let us say from option A to C what would be the correct answer. So, if we have more and more unsaturation, what do you mean by unsaturation? Unsaturation means presence of double bonds. So, unsaturation means presence of double bonds. Okay. So, presence of double bonds. So, I told you what will happen. Yes, so I have an answer option C. Lesser the unsaturation, higher will be the temperature. Yes. So, if we have double bonds, what is happening? kink formation was there. So if I have a straight alkyl chain, so let's say this is a fatty acid. Now if I introduce a double bond between here, let's say, what will happen, since the double bond has a cis formation, so what will happen, the double bond, because of the double bond there will be a kink. Okay, there will be a kink in the structure. Due to that kink in the structure, what is happening, that kink is making the structure more and more fragile. So greater the degree of unsaturation I will have, lesser the temperature, lower the temperature would be, or lesser the degree of unsaturation I will have, higher the temperature would be. So both A and C are the correct option here. Okay. So more and more unsaturation will be there, lower temperatures will be there before the gel, before the bilayer gels. So basically I have the membranes, right, lipid bilayer membrane. So if everything is straight alkyl chains, what will happen, the membrane will be very rigid. Due to introduction of such kinks, there will be some amount of fluidity. Okay, and it will require less temperature for it to become more and more fluid. Okay, so greater the number of unsaturation, lower the temperature, or lesser the number of unsaturation, higher would be the temperature before the gelling or the fluidity of the membrane starts increasing. Okay. So moving to question number 18. Which of the following molecules affect the mobility of the fatty acyl chains in the plasma membrane? Which of the following molecules affects the mobility of the fatty acyl chains in the plasma membrane? Option A, starch, option B, glycogen, option C, cholesterol, and option D, carbohydrates. So tell me what would be the answer. I have told you what affects the fluidity or the mobility of plasma membrane. Come on, we have discussed this question many times now. So what would be the answer? What is it that is present in the membrane that helps the membrane to become more and more fluid? Even at low temperature, the membrane is fluid because of which of the following molecules? I have told you this answer. What were we studying? Which structure we were studying? It is, yes, cholesterol, option C. So option C is the correct answer. So very good, Manisha. So cholesterol is present between the lipid molecules so that it can affect the mobility of the fatty acid chains when the temperature is changing. So at a low temperature, it prevents the membrane from becoming rigid. So it affects the mobility of the fatty acyl chains such that the membrane is always in a fluid state. Okay, it has some fluidity, some mobility present. Okay, so cholesterol, correct answer is C. Okay, so the next question, question number 19. Which molecule is most likely to diffuse across the cell membrane rapidly without any assistance? So which molecule is most likely to diffuse across the cell membrane rapidly without any assistance? Option A, water, option B, calcium Ca2 plus, and option C, RNA, and option D, nitric oxide or NO. So which of the molecules can diffuse across the cell membrane without any assistance? So what would be the correct answer here? Okay, we have an answer option A, water. Okay, anyone else wants to answer? Okay, let's see. So, if you remember what the membrane structure, what does it have? It has a polar head group and a non-polar fatty acid chain, right? So, it is a bilayer. So, for water to diffuse, can water diffuse without any assistance? Water is polar or non-polar? Tell me. Water is polar, right? So, if water is polar molecule, for it to move across the membrane, it will have to interact with non-polar fatty acid chains, which is not possible. So, water molecules will require some assistant, some carrier proteins that can carry those water molecules across the membrane. The only thing among all these, so calcium ions also, since these are also ionic, so they will also have some charge and they cannot interact with non-polar surface. So, water molecule, so water is polar, calcium ion is ionic, so it is also polar, so they cannot interact with non-polar environment, non-polar membrane, right? Nitric oxide, since it is gaseous and it is a non-polar molecule, it can easily, and it's a very small molecule, right? So, many factors are there. It is gaseous, it is non-polar, and it is very small in size. Okay. So, for nitric oxide, it is very easy to diffuse through the membrane. Okay, easy to diffuse without any assistance. So, it can diffuse on its own. Water can also diffuse, but it would be a facilitated diffusion. Water can diffuse, facilitated diffusion. What is facilitated diffusion? It is basically using a helper protein. Okay, any protein or any other carrier molecules, like something that holds its hand and take me out. So, if I want to get out of a room, if I am the water molecule and the room is the membrane, I need someone else to hold my hand and take me out of the room. Okay. Whereas nitric oxide, since it can walk on its own, it can diffuse out of the room on its own without any help. Okay, because nitric oxide is gaseous, non-polar and small in size. Why RNA cannot move out? Because of its bigger size. So, RNA is a big molecule, right? It's a huge molecule. So, for the big size also, it is difficult for it to pass through the membrane. Nitric oxide, it's very small, so it can easily pass through the membrane without any hindrance. Okay. So, that's why the correct answer is option D, nitric oxide. It can easily diffuse across the cell membrane rapidly without any assistance. Okay. So, next question, simplest hydrophilic moiety present in membrane lipid is, simplest hydrophilic moiety present in the membrane lipid is option A, phosphate group, option B, hydroxyl group, option C, amino group, and option D, glucose. What is the hydrophilic moiety present in the membrane lipids? It is mainly the hydroxyl groups. Okay, the OH, most of the polar head groups have this OH. Okay, this is the most hydrophilic and the most simple molecule that can be present. So, this hydrophilic OH, this is present in the membrane lipid. So, correct answer is option B. So, question number 21. Which of the following statement is true about the solubility of steroids in biological fluids? Option A, steroids and cholesterol are equally soluble. Option B, steroids are more soluble than cholesterols. Option C, cholesterols are more soluble than steroids, and option D, steroids and cholesterols are completely insoluble. So I've taught you the structure of cholesterol and steroids in the previous slide. So tell me what would be the solubility of steroids versus cholesterol. What was the difference between cholesterol and steroid? Anybody remembers? So if you remember the structure of cholesterol, it had the sterol nucleus. So what did cholesterol had? Cholesterol had a OH, then the sterol nucleus, and then alkyl chain, right? This was cholesterol. What does steroid have? Steroid lacks the alkyl chain, so no alkyl chain. And what is alkyl chain? Alkyl chain is hydrophobic, right? Alkyl chain, the alkyl chain that we have is basically hydrophobic, right? So if I remove a alkyl chain from the cholesterol, it is becoming less hydrophobic. That means steroids are more soluble than cholesterols. Okay, do you understand why? Why? Because steroids have a lesser hydrophobic part than the cholesterol. So sterol nucleus is also hydrophobic, but it has this polar head group which is hydrophilic. And if I am adding a more hydrophobic part, that is, I am making the molecule bulkier. Okay, so cholesterol will have a alkyl chain that is attached, that will make it more hydrophobic. The whole molecule will become more hydrophobic. When I remove this alkyl chain, it will become less hydrophobic. So steroid will be more soluble than cholesterol. So steroid will be less hydrophobic. Why less hydrophobic? Because I have removed the hydrophobic alkyl chain. Okay, steroids are less hydrophobic than cholesterol, so they are more soluble than cholesterols. Okay, so option B is the correct answer. Next question, question number 22. Which out of the following is not mediated transport? So which of the following is not a mediated transport? Facilitated diffusion, primary active transport, secondary active transport, and simple diffusion. Which of the following is not a mediated diffusion? So among these, simple diffusion is not a mediated diffusion. Why? Because if you see all the other options, facilitated diffusion, term facilitated means there is a helper protein or a carrier molecule. So helper is there. So it is mediated by a helper molecule. Okay. Primary and secondary active transport. The term active transport means energy. Okay. So active means there is a involvement of energy. So energy in form of ATP. So wherever active transport is happening, that means ATP is required. So something is required, so it is also mediated transport. This transport is mediated by ATP, that is giving energy for this transport to happen. Simple diffusion does not require anything. It is simply diffusing out of the membrane without any helper. So we studied the example of nitric oxide. So nitric oxide is actually doing the simple diffusion. Okay. So nitric oxide is doing a simple diffusion. Okay, this is clear. Next question is, which of the following is energy independent? Now I've told you again, active transport, primary active transport, secondary active transport, and passive transport. Which of the following is energy independent? Independent means does not require energy. So I've told you active transport requires energy in form of ATP. Okay, so if we have a membrane, so option D, passive transport, is the correct answer. Very good. So if we have a membrane and let's say there is a membrane protein, so there's something has to move from this side of the membrane to this side of the membrane. For this movement to happen, if it requires ATP, if it needs a hydrolysis of ATP into ADP and PI, so that means this is a active form of transport. Okay, active transport. For passive transport, it would be, so all these facilitated diffusion, they all come under passive transport. Okay, facilitative diffusion, simple diffusion, they will come under passive transport, where there is a carrier protein, there is no requirement of ATP, no ATP required. Okay. Okay, so passive transport. Correct answer is option D. Okay. Next question is, which of the following transports only one kind of substrate? Option A, uniport, option B, symport, option C, antiport, and option D, membrane proteins. Which of the following transports only one kind of substrate? What would be the answer? We have discussed uniport, symport, antiport carriers, so this is very simple. Come on, what would be the answer? Which of the following transports only one kind of substrate? Option A, yes. Very good, Manisha. So, option A, uniport carriers require only one type of one kind of substrate. So, I have discussed uni means one. So, that means only one substrate is moving from one side of the membrane to the other side of the membrane. For symport carriers, what is happening? Two molecules are moving simultaneously from one to the other side of the membrane. For antiport carriers, one molecule is moving from one side to the other side and second molecule is moving from in the opposite direction. Okay, anti means opposite direction. Okay, so uniport involves movement of only one kind of substrate. So option A. Okay, moving to the last question, which of the following induces conformational changes in the protein? So option A, uniport, option B, symport, option C, antiport, and option D, facilitated diffusion. Which of the following induces a conformational change in the protein? What would be the correct answer? So, a conformational change, yes, option D, very good. So, option D, facilitated diffusion, will induce a conformational change. How? So, let us say this is the carrier protein, this dark green colored is the carrier protein here. Okay. So, we have these molecules in the extracellular space that wants to come into the intracellular space. Okay. So, what will happen? A molecule will bind to this carrier protein. Once the molecule binds to the carrier protein, there is a conformational change, just like we saw it in enzymes, like the substrate was binding and there was a conformational change in the enzymes. Okay, similarly, when the molecules are binding to these carrier proteins, there is a conformational change and the molecule is basically, molecule was earlier open towards the extracellular space when the substrate that wanted to transport bound to it. Okay, so let's say there is a substrate that is there in the extracellular space. Now the carrier protein is open like this, the opening is towards the extracellular space. So the molecule will bind to the extracellular space and then at that moment there will be a conformational change and now the carrier protein will open towards the intracellular space, so that the substrate that is bound will be released to the intracellular space. Okay, so that's how facilitated diffusion is happening. So the diffusion is facilitated by these carrier proteins, the green colored proteins that are shown. The movement is facilitated by those carrier proteins. It is a type of diffusion only, and it requires a conformational change to occur in the carrier protein in order for the transport to occur. Okay. So this was the last question of the session. Any doubts in any of the questions, you can ask me. Anybody has any doubt? I hope everything was clear. So in this chapter, mainly you have to go through the structures of different kinds of lipids, the glycerophospholipids and the sphingolipids. What is the base structure of the sphingolipids and the glycerophospholipids? For glycerophospholipids, you have glycerols. For sphingolipids, you have sphingosine. Then the structure of different types of sterols, cholesterol, ergosterol. What is the base structure, the sterol nucleus, and then what is the role of cholesterol in the membrane, how it is affecting the fluidity of the membrane, about the potential difference across a membrane due to different concentrations of different substrates. Then you have the different types of transport. So all these topics are important in this session. So go through all the concepts. If we don't have any other doubt, we can end the session now. Okay. So thank you everyone for joining. I'll end the session now.